Okay, so in this lecture, we are going to have a look at the second stage of the processes which are happening inside your nephron. And you remember in the previous lecture, we looked at the ultrafiltration. That was the first step of a process. So today we're looking at the second stage, which is called selective reabsorption. I hope you remember it from last time. So selective reabsorption. So reabsorption. So just as a quick reminder, in the first stage, we took molecules, small molecules from your bloodstream, and those molecules were small enough to get through the little gaps in the Bowman's capsule, in the capillaries, and so as a result, they ended up inside that top part of the nephron. And so what you need to be doing at the stage of selective reabsorption, is kind of picking and choosing which substances you're going to keep and which substances you're going to leave inside the nephron and weave them out and therefore get rid of them. So the first step, the first question I'd like you to do, and you can do it from memory if you remember, but if you're really stuck, you can go back to your notes. So my question is, can you list for me the kind of a selection of small molecules which are going to be inside the nephron at the end of ultrafiltration. So list the molecules found in so-called glomerular filtrate, right? So this is the liquid which you ended up having inside your nephron, so found in glomerular filtrate. Okay, so at this point, I'd like you to kind of pause this video, gather your thoughts, and have a go at writing down a little list of everything. So these are the smaller molecules which manage to get from your bloodstream into the nephron. Okay. So I hope you didn't just skip and you actually paused the video and thought about it and looked up your notes. And if you're unsure, might be a good idea to go back and re-watch ultrafiltration lecture before you go any further. So some of those smaller molecules are going to include water. That's an easy one because we all know that's what you find inside your urine. So you also have this substance called urea. And again, if you're not sure where that comes from, then you should go and have a look at the introduction to urinary system PowerPoint, because it explains that kind of origin of urea in that PowerPoint. So it's water, urea, it's also glucose. So that's small enough to get through. It's also substances called amino acids, right? Which are building blocks of your proteins. And also you have things such as ions also included in that mix. So these are kind of the main kind of selection of the smaller molecules which make up glomerular filtrate. So now let's start looking at what's happening in the second stage, which is selective reabsorption. Well, in order to understand that, we need to have a go at drawing a nephron. Now, you can, of course, just go and copy and paste the picture from the internet, but I think it would be a good idea to have a go at drawing it with me, because as you're drawing a nephron, it really sort of makes you think about how these structures fit together, what the names of the structures are, etc. So I'm going to start by drawing the first kind of part of a nephron, and I hope you can remember what it's called from the ultrafiltration lecture. Again, if you can't quite remember, have a look, just pause the video and have a look and find it in your notes. So the name of that structure, that top of the chicken bone, right, the kind of dip, is Bowman's capsule. So Bowman's capsule. Again, just be careful, because that's something I'm looking for in the assignment, to make sure that you're spelling the structures correctly, and then that you're using capital letters in the appropriate places. So on the top of this Bowman's capsule, before I go any further, we have our network of capillaries. So I'm gonna use how do I close that? There. I'm going to use red color to show that network of capillaries. So we've got, you remember, slightly bigger blood vessel bringing blood in, then it splits in all this kind of network of capillaries, and then smaller blood vessel which is taking the blood out of Bowman's capsule. So again, as a reminder, let's remember what these structures are called. So the one which is bringing blood in, this is called afferent arteriole, and the one which is letting blood out of the system is called efferent arteriole. And you remember there was a reason why the afferent arteriole has got bigger diameter than efferent one. So you see I've drawn them in that particular way. So this one is wider than that one. So again, if you're not sure why that is, you should probably go back and have a look at the ultrafiltration lecture. So then that tangle of capillaries is called glomerulus. Glomerulus. Okay, and that's me kind of finished with my red color, so I'm going to go back to black color because I want to draw the rest of the nephron now. So from Bowman's capsule, we're going to enter the next structure called proximal convoluted tubule. And convoluted means like a bit like a maze, right? So lots of turns and, almost like a spirally kind of structure. So sometimes in some books it's drawn as a spiral, sometimes it's drawn as a kind of just folded tube. So maybe I'll do something, a bit of this and a bit of that. Okay, so almost like a snake-like structure. And then where I'm going to arrive next is a really kind of long bit which is going all the way down, and then it's going all the way up. So I'm carrying on with my snake-like nephron. So before I go any further, let me label this structure. So you remember this is called proximal convoluted tubule. So proximal convoluted tubule. Now you can appreciate this is a bit of a mouthful, so if you're a doctor which specializes in treating kidneys, you don't probably want to go around your whole day saying proximal convoluted tubule, get really fed up. So this is abbreviated to PCT. Right, then this kind of long loop is known as loop of Henle. So loop of Henle. And it's a little bit like Bowman's capsule. So Henle is a name of a scientist, so therefore capital H, please, right? Whereas the rest of this term, loop of, this is just all lowercase letters, unless it's obviously at the start of a sentence. So as this glomerular filtrate is traveling down proximal convoluted tubule, it's going to reach here, it's going to go down this way, and then it's going to go up this way. So this loop of Henle has got two so-called limbs, or two sections, if you like. Right, so let's figure out which one is which. So you've got ascending limb and descending limb. So which one is which, do you think? Right, so let's think about submarines. So if we're in a submarine, imagine that, and we are trying to descend. Are we going down? Are we going up? Right, so if you're descending, you're going down, and if you're ascending in your submarine, it means you're going back from the bottom of the sea to the top of the surface of the water. So if we now think about which direction this filtrate is traveling, so it's going down this way, and it's going up this way, so therefore that bit is going to be ascending limb, because you're going up, and this one is going to be descending limb. Descending limb. Okay, so but that's not it, because there is sort of further sections to my nephron. So what I'll try to do is I will need to erase some of this. So let me just get rid of this. How do I do that? How do I do that? Oh, here. Okay, that's better. So I just want a bit of space. A bit of space. Okay. Right, lovely. Lovely, lovely. Okay. That's a bit better. So why am I doing that? Because I need a bit of space to draw the next section. And this section is going to be so-called distal convoluted tubule. So I need to go here and carry on drawing. So the whole thing is going to kind of come up this way. Yes, I know, I know. You might think, well, I'm not very good at drawing. Well, give it a go. It doesn't have to be perfect, right? But I think it'll help you to just visualize those things. Okay, so if I was a bit better at planning my drawing, and don't forget I'm not using real pencil, so for me it's a bit trickier because it's all electronic pencil, so I can't sort of easily move stuff around. But this is descending limb. Oh, not descending, sorry, distal, where is my brain? Distal convoluted tubule, which is again, because of how long this term is, this is abbreviated to DCT. Okay. And so finally, let me just do a slightly better job with that. I'm not terribly happy with that. So let me do, is that going to allow me to do eraser? Okay, let me just do a slightly better job. Have another go. Okay, so I'm going round and there. Okay, and at this point you're gonna arrive finally to the last part of your nephron, and this last part is called collecting duct. Collecting duct, right? So you can see that DCT is going to be taking the remaining filtrate into the collecting duct, right? So if I just keep on to the point having these arrows, so we're going up, along, and then down this way and into collecting duct. So those other kind of branches, if you like, they are bringing a bit of this glomerular filtrate, which is essentially at this point is urine. This is what's, they're bringing that remaining bit of glomerular filtrate from our nephrons. So imagine have a bunch of those nephrons all collecting a little tiny bit of fluid, a tiny bit of remaining filtrate into collecting duct. So this is from another nephron. Okay, perfect. Right, so I know it's quite a kind of long and big and complicated drawing, but I think it'll help you to kind of visualize the process and really relate what you're looking at and what you're reading in books to the theory. So let's just recap. So that bit of a process with afferent arterial glomerulus, efferent arterial Bowman's capsule, all of this is involved in this first step, which is ultrafiltration. So the rest of your nephron, so this is PCT, loop of Henle, DCT, and collecting duct, all of this remaining section, all of the remaining section of the nephron is dedicated to selective reabsorption. So why do you have such a long nephron all dedicated to this process of reabsorption? Well, I hope some of you are able to figure it out, yourselves. It's essentially, you want to give the useful substances as many chances as you possibly can to go back into your bloodstream and be used by your body. So if the nephron was really short, you just wouldn't have enough opportunity for these substances to get back into the bloodstream. So it's a bit like what's happening with your digestive tract. You want your small intestine, where you have absorption happening, to be, long, right? So you're getting all these useful substances into your bloodstream. So this is kind of similar. So you want nephron also to be long, and make sure that everything you're wanting to reabsorb is getting reabsorbed. So let's now start talking about sort of individual structures. So I'm going to start with PCT, because that's where we're arriving first. So PCT. So what's happening there? So to understand that, we are going to have a go to another drawing. But in essence, PCT is where majority of the substances are going to get reabsorbed. So majority of reabsorption takes place. Okay. So that includes, so all these substances we wrote down earlier on. So most of your water, most of your glucose, most of your amino acids, and a lot of the ions are reabsorbed there. I'm going to let you figure out or look up or research yourselves specific percentages or specific figures to go with each substance. How much water, for example, gets reabsorbed in PCT specifically. So I'm going to leave that to you to kind of find out. So what I'll do next is, if I have a section of PCT, so imagine I took a little section from here, and I'm going to enlarge it to see what's going on inside PCT. So imagine that's my tubule section of PCT. So I'm going to have capillaries running alongside PCT on both sides, actually, because they're sort of swirling around the nephron, and they're wrapping around nephron, so absorption can take on both sides. So I could do it like that, I suppose, just to clarify. Try to imagine this is capillary and this is another capillary. So it's capillary. This is capillary on the other side. And between these two black lines, that's where your PCT is. So inside PCT, what you're going to find is that it's lined with these specialized cells, and I'm going to draw a few, right? And then I'm going to again enlarge them. So imagine these are the cells which are lining the walls of PCT. So I'm going to draw a few here and a few on this side. Okay. So let's say another one there. Okay, right? So, and etc. etc. So I'm just not going to draw too many. So it's just going to carry on in both directions, right? So you can imagine, so inside here, inside this space, that's where filtrate is going to be. So let's try to maybe color code it a little bit. So if we say that filtrate is going to be in green, so this is glomerular filtrate is here in this area, and then between that sort of space, so maybe I'll do it in, I don't know which color shall I use, maybe, I don't know, blue, for example, right? So in this space you will have just fluid, right? So fluid, so-called interstitial fluid, right? Interstitial fluid. So this is basically, just fluid between the cells in your body. So what you're essentially trying to achieve, you're trying to move substances from glomerular filtrate back into capillary. So they can go that way, or those substances can go into capillary on the other side. Okay, so in essence, those smaller molecules are something like water or glucose, so it needs to get inside that cell, which is lining PCT. It needs to get out of this cell into this space filled with fluid, and then it needs to get through the wall of a capillary in order to end up back in your bloodstream. So that's the process which needs to happen in order for reabsorption to take place. So now what I'm going to do, I'm going to enlarge one of these cells. So imagine, let's pick, for example, this one on this side. So I'm going to just draw it bigger. So that line is there. So all I'm doing is just zooming in, zoom in. And here, imagine that's one of these cells, but now I'm going to draw it a lot bigger. Okay, so something like that. Okay, now so let's think together what's what. So in the middle of that cell I've got a blob. What do you think this blob is? I hope everybody is able to tell me what it is. Okay, and also I have these kind of little projections, and I hope you kind of remember that we already came across cells with projections like this in a different assignment, in a different unit, and they were cilia, but in respiratory tract. But this time these projections, even though they're shaped pretty much the same, but they're actually called microvilli. So they're called microvilli. Okay, so microvilli are many. Okay, so microvilli. So have a look, label a couple of them. So that's one of the features of this cell is microvilli. Another big feature of this cell are presence of a lot of organelles which are shaped like that. And again, I'm really hoping that you should be able to recognize what I'm drawing here. So a lot of these elongated oval-shaped organelles with folds inside them. Okay, so let's now label a few of these things. So I hope that middle blob you understand what that is. So this is your nucleus. And I'm also hopeful that you're able to recognize these organelles. So they are mitochondria. So if you have correct answers in mind, then very, very well done. So next I'm going to ask another couple of questions. So question 1, why do you think these cells have microvilli? What do you think these microvilli are doing? Now, if you think they're doing the same job as cilia, sorry, but you're not quite correct. So can you think of a different reason rather than cilia? So remember, cilia in the respiratory system was kind of moving mucus along. So they don't need to move anything along, but they're there for another reason. So why do you have microvilli? And why do you think these cells in PCT have a ton of mitochondria? What do you think these are doing? Okay, so again, I'd like you to pause the video and have a good think about that, and maybe write down your kind of suggestions for the presence of microvilli and mitochondria in these cells, and then restart the video again. Okay, so let's try to think about the answers. So first of all, the reason for microvilli is larger surface area. So if that's something you had in mind, then very, very well done. So why do I need larger surface area? You remember what I'm trying to achieve here is move as much substance across as I possibly can. And so therefore the bigger surface area you have, right? So it's not just the length of the nephron, that's going to provide bigger surface area. But that's not enough. So as well as just the sheer length of a nephron, you also have a lot of cells which have got these extra folds to provide even bigger surface area. So, and then the reason for mitochondria. Well, let's try to remember from cell biology what the function of mitochondria. And the main function of mitochondria is to produce energy. And here we need to start scratching heads and think about, well, okay, why do I need to have energy a lot of energy in these cells? So where is this energy going to go? What is it being used for? And again, you need to think back to cell biology assignment and think about what are the mechanisms by which substances are transported across membrane, because that's what I'm doing. I'm transporting it across those membranes into the cell, then across membrane into the space, interstitial space, then across another membrane into the capillary. And the two main um, mechanism by which those substances are going to be transported are, first of all, diffusion. So let me write it down. So diffusion is one of them, but not just diffusion, but another mechanism which is going to be at play here is called active transport. Right? And if you remember, diffusion is the mechanism where you do not need energy because you're moving from high concentration to low concentration, but you're also going to have active transport, which moves substances from low concentration to high, and that does need energy. And because you're using active transport to transport those substances, such as glucose, for example, or amino acids, you will need to have mitochondria for that purpose, right? So that requires ATP. So my suggestion would be, when you're describing what's happening in PCT, remember that's where we are at the moment. We're inside PCT, we're thinking about how those substances are getting transported. So when you're describing what's happening in PCT, make sure you're clearly describing what diffusion is and what active transport is. Now, you probably already described it in your cell biology assignment. So I don't mind you using those descriptions and incorporating them into this urinary assignment. Just put in brackets after the paragraph you kind of copied from your cell biology, taken from my cell biology assignment. So then I know that that's the reason why similarity might be a little bit on the high side. Okay? But what I would suggest, just make sure your explanation flows well. So you don't just sort of chuck that paragraph in, but there is no connection between the paragraph about diffusion to the previous explanation or the kind of a paragraph after it. So make sure it kind of flows logically and everything makes sense for your reader. Okay, so that's what's going on in PCT. So let's just go back up again and have a look where we are. So that's where we ended up. So a lot of our water and glucose and amino acids are going to get reabsorbed here. So this stuff is going to move from inside, from glomerular filtrate into capillaries. So now we need to start discussing what's happening in the loop of Henle. So describe that. I'm going to have a go at drawing yet another diagram. So let me just try to go into the next page if I'm allowed to in this Word document. Let me just go. Okay, good. So next page. So on this diagram, I'm just going to purely draw loop of Henle. So I'm not going to repeat drawing of PCT and DCT, etc. So I'm just highlighting what's happening inside the loop of Henle. Let me just try to make sure I'm in the right setting for the drawing. See if that works. Does it work? Why doesn't it work? Problem with technology, isn't it? There's always something. Okay. Just make sure I've got plenty of space. Come on, draw. Draw. Ah, okay, good. Back in business. So, loop of Henle then. Okay. And there we go. Right. Okay, so let me just remind you first of all. So you remember that this is going to be my ascending limb. You remember, because the fluid is glomerular filtrate is going up. So ascending limb. And this one is descending limb. Yeah, my pencil is not cooperating now. Okay, so that's going to go down. Okay, so here is the thing. So in the ascending limb, that's what you need to kind of describe in your assignment. This limb is impermeable to water. So impermeable. Okay, let me just try to fix this pencil. Before I go any further, all I need to do is just draw here, and hopefully that should fix it. Okay, try again. to water. And this one is impermeable to ions. So impermeable to ions. Okay, so what's happening here is, and this is, pretty complicated process, you've got to kind of pay attention, is we're going to start kind of describing it from this ascending limb. So here what's happening is you've got two main ions being pumped out of this ascending limb into this space. And these ions are ion of sodium, and it would be interesting to see if you know how to write it down as a chemical formula. So sodium is Na, and it's an ion, so it's not just an atom, so it's not, Na is just not enough. I need to also show that it's a positively charged particle, so it's atom of iron which lost one electron. You remember electron is negative, so therefore that's what is leaving that positive charge behind. And another ion which is going to get pumped out here is chloride ion. So you've got a whole bunch of these sodium and chloride ions being pumped out. So what does that do? Well, what you have to think about is that your sodium and chloride together, that makes a substance which you all can find lying around in your kitchen. And this is essentially table salt. So this is table salt. Right? So therefore, what you have here is a very salty environment. And by creating this very salty environment, what you're doing in essence is you're drawing water out of descending limb. So that very salty environment is going to start acting on descending limb, and it's going to be drawing water out of it. And water is getting drawn out of descending limb through a process called osmosis. So this is just a property, if you like, of how sort of salty environment acts on water. So just to give you a bit of kind of analogy, there is a pretty straightforward experiment where you take some chunks of potato and you measure their size and weight, and you put those chunks of potato into very, very salty water, and then you leave them. And what's going to happen, and it's going to happen pretty quickly, I mean, you can leave them for like 10, maybe 15 minutes, and then take them out of this salty solution, dry them off, and then weigh them and measure them again. And what's going to happen to those chunks of potato is that they're going to actually shrink. And the reason why they're shrinking is because that water is drawing, because the salt is drawing that water out of potato cells. So kind of similar thing is happening here. Because you've got this very salty environment, that's what's going to be drawing that water out of that descending limb. Because what you can't do with water, you can't actively transport it. There is no system for your body to actively transport water. So therefore you have to kind of make it go where you want it to go, right? And one way of doing it is creating this salty environment so the water kind of follows, and that's what's going on with the loop of Henle. So this particular mechanism has got a name to it. I'm afraid it's another mouthful. It's called countercurrent multiplication. So that mechanism of how you're drawing water out of loop of Henle is countercurrent. Let me try to write it down. Countercurrent multiplication. My pen is not working very well here. Multiplication. Well, I had to go. If you're unsure about spelling, just Google it. Also on VLE there is a really good video if you feel you haven't quite understood it well enough, or you want some more information, there is a Khan Academy video, and there is a link to that video on VLE. So by all means, have a look at that. And so finally, once you've left loop of Henle, you are going to arrive to distal convoluted tubule and collecting duct. So I'm not going to draw them again, but essentially at this point, when you arrive to distal convoluted tubule, you only have around about 10 to 15% of your filtrate remaining. So absolute majority of around about 90% or just under got reabsorbed in the proximal convoluted tubule and loop of Henle. So we're really playing with this kind of remaining last 10% of a filtrate. And this is the point where your body, if you like, will decide, or kidneys decide, in inverted commas, of course, whether you're going to reabsorb a bit more of the water or a bit less. So anything before that, you reabsorb regardless, right? Whether you drank recently, whether you drank a while ago, 90% of the water in the nephron is going to get reabsorbed anyway. But you have this kind of remaining 10 or 15% to play with, where your body then kind of fine tunes how much of this water should become urine, how much of this kind of filtrate should become urine, and how much of the remaining water should be reabsorbed. So obviously, if you drank recently, you want to most of this remaining 10 or 15% to stay inside the distal convoluted tubule, inside collecting duct, and become urine. If you haven't drank for a while and you're thirsty, you want to retain more of this water, and therefore more of that water is going to be reabsorbed. So the hormone which does that job of reabsorbing water is called antidiuretic hormone. So let me write it down somewhere. And so it's abbreviated to ADH. So ADH. So antidiuretic hormone. So this is the hormone which is produced in your hypothalamus, in your brain, and that then hypothalamus kind of essentially collects in your pituitary gland, and that's where it stays. And then your pituitary gland releases ADH, so it goes into your bloodstream, and it acts as any hormone. You remember, you have to have lock and key mechanism here. So the receptors for ADH are located in the cells of distal convoluted tubule and collecting duct. So what ADH does, it kind of makes distal convoluted tubule and collecting duct more permeable to water. So therefore more water is able to leave. So you have more ADH, more water is able to leave. DCT and collecting duct. So here I want you to kind of have a think about a couple of scenarios. So if you drank quite a lot of water recently, do you want more ADH circling around your bloodstream, or do you want less ADH? So remember, ADH makes those tubules more permeable. So makes, let me just write it down if I can, makes DCT and collecting duct more permeable. Okay, so what I want you to think, so knowing that information, so if you drank a lot of water recently, do you want more ADH or less ADH? And on the other hand, if you haven't drank for a while and you're thirsty, how much ADH do you want in that particular scenario? So I want you to kind of have a think about it. And also I'd like you to have a go at drawing kind of a couple of loops, homeostatic loops, to describe that process. So if you start with a normal level of water and then you haven't drunk for a while, what's going to happen? What's going to be the chain of events in that particular case? Okay, and finally, just to finish things off, I wanted to show you a couple of pictures. So let me just go into this PowerPoint. It'll be easier if I do it using PowerPoint. There it is. Right, so the first picture I wanted to show you is a diagram of a nephron. Right, so this is what we were trying to draw. So you can see Bowman's capsule here and your afferent and efferent arteriole, and you can see how it's then branches off into all these smaller capillaries which are creating network around the whole of the nephron. So that's where the water and all the other substances are going to get reabsorbed. So proximal convoluted tubule here you're going into loop of Henle, going down, going up into distal convoluted tubule, and finally into collecting duct. And that's what I was talking about, where you've got these other kind of branches of collecting duct, which are essentially where other distal convoluted tubules are going to be connected up from other nephrons into the same duct. So here is a picture of the cells which we were drawing inside proximal convoluted tubule. So you have this microvilli, those projections, and here you can see plenty of mitochondria, and that's the nucleus, by the way, in that cell. And then the next picture I wanted to show you, so well, how does this relate to the actual kidney? So if you have kidney here and you take like a little slice out of kidney, the inner section of kidney is called medulla and the outer shell is called cortex. And so you can see that your nephron is kind of sitting inside both cortex and medulla. So the upper section of a nephron, so will be Bowman's capsule, DCT, and some of the collecting duct, so kind of that section is going to be inside cortex, that bit, right, closer to the kind of the edge of the kidney, and then loops of Henle and sort of deeper sections of the collecting duct are going to be inside medulla. Okay, so all these collecting ducts are going to be collecting tiny bit of urine, and that's going to end up in this section called pelvis, and ultimately it's going to come into your bladder and out of your body. And finally, just to relate what I was saying about hypothalamus and pituitary gland, where they are. So they're both located inside your brain. So that would be the front of your face here. That's the back of your head. So really deep inside your brain, you have these two structures, hypothalamus there, and then this gland called pituitary gland on a little stalk kind of sticking out. It's encased actually in the bone, so it's very well protected by the surrounding bone. And this is pituitary gland. So your ADH is going to be produced in hypothalamus, then it's going to migrate into pituitary gland. It's going to be sitting there waiting to be released depending on the circumstances. So that pretty much completes the lecture on selective reabsorption. I appreciate this is a complex topic, but you should be familiar with most of the processes. So if you understand the process of diffusion, active transport, osmosis, then you should be able to follow that explanation and do a bit of your own independent research as well.